Oh, after your physics discussion? Oh, yeah. I'll have a, no, I have a philosophy thing that I use in my philosophy. Like, I have to work right after. Yeah, but it's 150. Oh, wait, no. So you can finish your thing. There you go.
Okay, it's 1:30. Let's get started. It's just reminded, like a couple years ago, teaching during the pandemic, and everybody was sitting far apart, and nobody said anything. You know, it's so nice that I have to tell you to quiet down. So I love that. So looking ahead, our next exam is 2 weeks from today. So not this week, not next week. We got a little space yet, but it's kind of on the horizon. So we'll cover more material. So try to kind of keep up. I know a lot of you are, but probably some of you aren't. We did a lot last time. We went through a lot of pathways. So are there questions from last time? Anything we need to clarify as we move forward? We ended at a good time. So we're kind of fresh for the electron transport chain, I think. But when we ended last time, we were looking at this kind of variation of the Krebs cycle called the glyoxylate shunt. And I assume that's probably new for most of you because it's not something that we do as humans. Remember, this is only something that most bacteria can do. There's some fungi and plants and other organisms that can do it too. But remember, it's just we're cutting off part of that Krebs cycle. Some bacteria will do this because you can see there's a couple carbon dioxides, carbons that they're losing there. And if they have an enzyme, a specific enzyme that allows them to kind of cut that off, then they can conserve those carbons. They are added together with another acetyl-CoA to make an additional malate, not just the one that you get from the Krebs cycle, but an additional one. And then that can be used to make lots of different cellular structures. So that's sort of the thrust of what we're talking about. Some bacteria then can be a little bit more flexible in terms of they don't have to use a sugar. They can use some kind of 2-carbon compound, not necessarily just glucose as an energy source. Okay, so let me give you a couple other practice questions here, looking back before we look forward. Take a minute and choose the best answer there. All right, so let's go through each one here and decide why it's true or why it's not true. So which of these is true about the Entner-Dodorf pathway? Is it another name for glycolysis? No, but the naming is kind of confusing there. Remember, it's Embden-Meyerhoff-Parnas glycolysis, which is kind of what we normally call is just glycolysis. So kind of sounds similar. So that's not correct. Is common to most forms of life? No, again, that was Embden-Meyerhoff-Parnas. This is only some bacteria that are able to do this pathway. Produces half the ATP as glycolysis. Yes, that's the correct answer. Is an anabolic pathway? No, we're still getting ATP out of it. So that's a tip that it's a catabolic pathway. It's breaking molecules down, not building molecules up. It's carried out at the same time as glycolysis. No, organisms are either doing Embden-Meyerhof, Parnas, glycolytic pathway or that Nerdodorov pathway. Not typically, as far as I know, ever doing them both at the same time. Now that other glycolytic pathway that we talked about, pentose phosphate, yes, organisms can be doing that at the same time as glycolysis, or yes, can be doing that at the same time as Nerdodorov. Okay, one more. I know we haven't talked about fermentation yet, so hint, that's not the right answer. So the electrons that drive biosynthetic reactions, those are going to be carried by NADP. Okay, and those are going to be used in the pentose phosphate pathway, right? Also in the Nerdodorov, that wasn't one of the possible answers there. Okay, both of these are having NAD, and that gets carried when it carries electrons. It's NADH, of course. The Krebs cycle also has the FADH2 and the NADH. One other thing that I wanted to make a little correction, and I did post this under the tab on Blackboard for learning objectives. I made some tweaks in between semester, and I forgot to translate a few of those tweaks to the learning objectives. So and again, I have this posted, so you don't need to try to hunt that up now, but I added the term amphibolic instead of just catabolic and anabolic. Remember that a lot of these pathways can kind of go in both directions. And this second learning objective, basically, we talked about those terms like autotroph and heterotroph with the last chapter, with chapter 7. I used to talk about it here, and I dropped a couple terms, organotroph and lithotroph, but you don't have to worry about those. So whenever I kind of add a little something in one spot, I try to simplify something somewhere else. So those were the only two things that are changed. So just make you aware of that. Okay, so moving forward in aerobic respiration. Okay, so this is our overview. We just got done talking about the Krebs cycle. We have these electrons that are coming off the Krebs cycle being carried by NAD, reduced NADH, and FAD, reduced FADH. I said before, it's all about the electrons. It's those electrons that's going to be changed into lots of ATP. Now, we did get some ATP coming off the Krebs cycle by a substrate level phosphorylation, and same way some coming off glycolysis. But it's those electrons that are going to translate into a lot of ATP at the electron transport chain. Okay, so this is a series of redox reactions that are always going to take place in some membrane. Okay, so there's a couple different ways, couple different graphics I want to show you here. One's better at explaining certain things and one's better at explaining other things. So I want to show it to you a couple different ways. Electron transport system. So like I said, this is going to be in the plasma membrane. I don't know if I said that or not, of prokaryotes. And the inner mitochondrial membrane for eukaryotes. Okay, so remember that mitochondria has a cell membrane or a plasma membrane, and then this other membrane inside there. Okay, it's really important that that membrane is intact, and I'll show you why pretty soon. But so basically what the electron transport system or electron transport chain it's a series of electron carriers that are there in that embedded in that membrane. And so here's some examples of electron carriers. And you don't need to memorize the order of these. Some organisms will have different carriers than other organisms, but this is just kind of an example of the kinds of molecules that we see. We see flavoproteins, we see cytochromes, quinones, iron sulfur proteins. These are all molecules that can switch really easily from an oxidized form to a reduced form. Okay, so they can switch back and forth. So to begin with here's our NADH. Remember, it's carrying a proton and 2 electrons, call that hydride ion. And it's going to drop off that proton and 2 electrons at this first carrier. It happens to be flavomonoprotein, FMN. I forget what that stands for. I have it written somewhere. Flavin mononucleotide, I believe. Okay, so NADH oxidized to NAD. Okay, that's where the term oxidative phosphorylation comes in. As we'll see, there is some final electron acceptor that's absolutely necessary, but it doesn't have to be oxygen when we're talking about bacteria. Okay, so the NADH is oxidized, it's dropping off its electrons, and then these different carriers, again, they alternate between the reduced and oxidized state. They're passing the electron down. Okay, the reason that it's drawn in a diagonal like that is because that's the energetically favorable way for those electrons to be passed. So it's like, you know, you put a ball at the top of a hill and it's going to roll down. And as those electrons are passed, the protons get pumped. This is outside the cell. Again, assuming we're talking about a bacteria here. Okay, so the protons get pumped outside of the cell. Okay, and that's going to create a gradient. We call that a proton gradient. Okay, so we'll just say proton gradients. More outside of the cell than inside of the cell. That's going to be important. You'll see that FADH2 drops its electrons off a little bit further down the hill, not quite at the same spot. So those electrons from FADH actually converts into about 2 ATP per electron. Okay, so probably should write that in here somewhere. This is fine. So 1 FADH2 is going to convert into about 2 ATP, and 1 NADH is going to convert into about 3 ATP. Again, because here the NADHs are dropped at, say, the top of the hill, and the FADH, the electrons are a little bit further down. So here, you see the ATPs in yellow. That's a little bit confusing because they're not created right there. As we'll see, there's one enzyme complex kind of at the end of the pathway where they're created. But this is showing about, well, there's about enough electrons here that would convert into an ATP. But again, that's a little bit confusing. Okay. Let's see, what else do I want to say? The cytochromes. If this final electron acceptor is oxygen, this final cytochrome here, we always call this cytochrome oxidase. So that's kind of an important one. The final one, again, if this is aerobic respiration, we call that cytochrome oxidase. And there's a couple different variations of cytochrome oxidase. So that's a test that we can do in the lab. There's 2 huge families of bacteria one that makes one cytochrome and one huge family that makes a different cytochrome. So that's one way we can use to kind of tell certain big families of bacteria apart. Okay, so keeping all that in mind, here's the figure from your textbook that's a little bit better at showing some other things. So it doesn't show as many electron carriers there. That's okay. Here's outside and inside the cytoplasm inside the cell. Notice here, this shows oxygen as being the final electron acceptor. And that you can see two protons combine with half an oxygen, half an O2. So that O2 molecule has to split in order to accept those electrons and end up as oxygen. That splitting doesn't always happen correctly. Somewhere maybe about 2% of this time we get superoxide instead of water. Okay, so remember we talked about superoxide. That was one of those reactive oxygen species or toxic oxygen species that it's just kind of a byproduct of aerobic respiration. Sometimes some of the electrons will kind of leak into the cell. They don't get passed, they get fumbled basically as they're passed to those electron acceptors. And that can cause some superoxide formation as well. So especially cells that are respiring aerobically, and really any cell that is around other organisms that are respiring aerobically have to be able to detoxify that superoxide. Otherwise, they're not going to be able to survive. Okay, so that's a little bit about toxic oxygen. And here in this picture, they show you the ATP synthase. Okay, that's the one spot where I said that we had this proton gradient, lots of protons being pumped outside of the cell. That's the one spot that those protons are allowed to diffuse back in. We actually call it chemioosmosis. Okay, that's when those hydrogens, those protons can diffuse in down their concentration gradient from a high concentration to low concentration. And that process of them doing that causes the phosphorylation of ADP to ATP. Okay, so what started out as the NADH being oxidized to NAD ends up in this phosphorylation. Okay, so I used a couple of terms. I used said that there's a proton gradient. I used this term chemioosmosis. This slide kind of teases those terms apart a little bit. And the other term, I don't know if I mentioned this term, that proton motive force, because there's a proton gradient, that's actually potential energy outside of the cell. We call that the proton motive force. And when, again, it diffuses in down the concentration gradient, we call that chemiosmosis. That causes the phosphorylation of ADP to ATP. So those two terms, chemiosmosis and proton motive force, are kind of confusing. Basically, because there's this potential energy, this proton motive force, then chemiosmosis happens, basically. Okay, another great illustration of this concept. I didn't post this picture. I forget which dam this is. I knew at one time it might be that one in Las Vegas, near Las Vegas. I forget. Has anybody been there? Yeah. Is it Grand Coulee Dam or is it Hoover Dam? Hoover Dam. That's it. Okay. So if the dam is the cell membrane, right? You've got an intact cell membrane. Okay, those protons can only diffuse back in for the cell membrane, right? That enzyme ATP synthase. Okay, just like the water can only flow through at one spot where the turbines are. And that's how we get electricity from the movement of water. Okay, think about what if there were some holes in the dam? Okay, water would be coming through at other spots and not getting any electricity for us. Not to mention that would compromise the integrity for sure of the dam. But we can't have lots of little holes in the cell wall. We can't have the protons coming through lots of other places. Otherwise, the ATP is not going to be generated. So we have some antibiotics that will actually poke holes in cell membranes. Problem is that our cell membranes are very, very similar to bacteria cell membranes. So those drugs are generally pretty toxic for us as well. We have to be very careful how we use them, like maybe only topically on our skin. So, okay. All right, so this figure is kind of a nice little summary of aerobic respiration. And it talks about some totals here, maybe a little different way than what I have seen them summarized before. And I'm not too worried about numbers here, but if you kind of get the gist of, oh, a lot here and not very much here. So during glycolysis, you're going to get, we end up with 2 ATP by substrate level phosphorylation. And then because we also get 2 NADH during oxidative phosphorylation at the electron transport chains, those 2 NADH, a 3 for each NADH will translate into 6 ATP. So theoretically, from events of Embden-Meyerhoff-Parnas glycolysis, we would get 8 ATP. There's a little transition reaction there where we got a couple of NADHs as those pyruvates were changed into acetyl-CoA. And then from the Krebs cycle, because we're getting so much reducing power here, it's all about the electrons, those will translate into a lot of ATP, 24 from the Krebs cycle. Okay, so totals, theoretically, about 38 ATP from bacteria. Make a note here just that for eukaryotes, theoretical total is going to be a little bit less, 36 total, because we have to spend an ATP to transfer the NADHs from glycolysis into the mitochondria. Okay, so just note that for eukaryotes, that theoretical total is slightly less. But even for bacteria, I emphasize theoretical because bacteria will use that proton motive force that's generated for other things besides making ATP. Bacteria, again, use that proton motive force for a couple things. One, to move their flagella. Okay, they're not spending ATP every time they spin their flagella around. It's the proton motive force that powers that. And they can also use the proton motive force to transfer things in and out of the cell, just to make an active transport, just to make it a little bit more favorable. So that theoretical number is probably not ever reached, or rarely. Okay, so we were speaking primarily to aerobic respiration. What are some differences with anaerobic respiration? It's a lot of similarities. The glucose is going through glycolysis pathway and the Krebs cycle. That's only modified a little bit and a little bit of a modification of electron transport chain. And then you have some electron acceptor other than oxygen. It's usually going to be an inorganic ion. Okay. If it's organic, it's always something that's come from outside of the cell. Okay, and that's not very common, but it could be an organic electron acceptor just from outside of the cell, not something that's already inside the cell. Okay, so what are some examples of, and let me just emphasize that it's only prokaryotes that are able to do anaerobic respiration. Obviously, we don't do this. Okay, so what are some examples of some inorganic ion? It could be nitrate, used as an inorganic ion. So let me write that reaction because this is one that you're going to talk about in lab next week. If you add a few protons and a couple electrons, and the organism can produce an enzyme called nitrate reductase. Okay, then we end up with nitrite in water. I know that's cumbersome to type that out, those of you who are typing. And not that you have to memorize that reaction. Again, this is one that you'll talk about in lab a little bit. And it's a little bit harder to understand why nitrate is reduced to nitrite. You have to understand like oxidation states in order to assign that. But generally, when an oxygen is lost, that's a reduction most often. This enzyme, nitrate reductase, I can't remember if I posted this or not, this picture, but essentially this is an enzyme that's right there in the cell membrane, accepting that enables the organism to use nitrate to accept those electrons, you know, instead of using oxygen. Okay, so nitrate reductase is something that we can't manufacture. Some other examples of final electron acceptors, something like sulfate. And here, hydrogen sulfide is the end product. We are in lab going to be testing for the presence of hydrogen sulfide way at the end of the semester. Hydrogen sulfide is that gas that, like, if you've ever been to Yellowstone, that smells like rotten eggs, that really sulfury, nasty smell. Okay, that's a bacterial as a result of some anaerobic respiration done by bacteria. Carbonates or sometimes carbon dioxide can also be an inorganic ion that's accepting electrons. Here's an example of some organisms that can convert carbonates into methane. Okay, and there are organisms in the domain archaea that can do this. They're called methanogens. Okay, they actually produce methane. So their presence in like the rumen of cows, for example, you know, cows could release methane. Whoop, forgot the H there, methane, sorry. Okay, so here's an example how we can utilize organisms that make methane. Of course, in landfills, we're constantly, you know, covering up trash, lots of organic material. So there are anaerobic organisms that live, you know, deep in landfills. We have to have that vented properly because they're producing gases as a part, as an end product of their respiration. We can collect that gas and use it for electricity. Now, right now, this is done on really small scale. I know the landfill just south of Grand Rapids does this, collects kind of enough to power their little office, whatever. It's not really economically very feasible yet, but it's kind of a cool idea that, you know, we can produce electricity from the action of microbes. So again, back to this kind of overview reaction. If the organisms here, or this overview slide, again, this final electron acceptor doesn't need to be oxygen. It can be something else. It's still the same pathway, anaerobic respiration. There's a Krebs cycle. There's an electron transport chain. We're going to get to fermentation in a minute and how that's different. Fermentation doesn't require oxygen either, but it's a very different process. There's one more point that I want to make about anaerobic respiration. And bear with me here a little bit as I show you this complicated looking redox tower. But the point that I want to make is that organisms that do respire anaerobically, they always get less less ATP than organisms that will respire using oxygen as a final electron acceptor. Okay, so less ATP is produced from anaerobic respiration. Compared to aerobic. And I could just tell you that and have you memorize it, but I think it's helpful sometimes to understand why that is. It might stick with you a little bit better. So we can actually put electrodes in cells and measure this redox potential. And we measure that in something called in volts, but something that we call E naught prime. So that E0 prime, we pronounce that E naught prime. And basically, if that voltage is very negative, that means some molecule really wants to give up electrons. Okay, so here, very negative, those molecules really want to get rid of their electrons. If E naught prime is positive, then those molecules really want to gain electrons. So if you think about in aerobic respiration, right, we had the NADHs dropping off their electrons. Here's NAD, that NAD-NADH pair. Okay, it has a pretty negative E naught prime. So those NADHs want to get rid of those electrons really quickly. And then what's accepting it right here, here's the oxygen, half oxygen, accept those electrons to make water. That is a really positive E naught prime. So it wants to gain electrons. So when you measure the distance between those two, that enables you to calculate the free energy. Okay, we're not going to do that. I'm going to leave that for your biochemistry professors to calculate the free energy, but it's the distance over here. I said the greatest change in reduction potentials is going to produce the most energy. This is a pretty long distance. Say something other than oxygen is used to accept electrons. No, I didn't remember to do it. Okay. Say something other than oxygen is used, like let's see, here's sulfate. Okay, well, that's not a very large distance, so that organism is not going to gain a whole lot of ATP from those electrons. Here's nitrate down here. Well, that's a little bit further, and that nitrate reduction can actually take place in a couple of different steps. So here's if it was kind of a fuller reduction. Okay, so whether sulfate or nitrate, it's not as far. You're not going to get as much ATP there. And here, this also enables us to understand the order of electron carriers in there, as to how easily those electron carriers, like cytochromes or ubiquinones or whatever, are giving up their electrons. Okay, so what's the advantage to the organism if it's not making much ATP or as much, still making more than some fermentative pathways we'll get to in a minute? What's advantage to the cell using anaerobic respiration? Where can it live? Places where there's no oxygen, right? Okay, but enables the organism to live where there's no oxygen. So, deep within a landfill or some swampy area. Okay. All right, so what kind of organisms are respiring aerobically? We talked about oxygen requirements last week. Facultative anaerobes. Those are organisms that can use oxygen, or they can do some kind of metabolism without oxygen. Aerotolerant anaerobes are probably either doing anaerobic respiration or fermentation. Strict anaerobes, either anaerobic respiration or fermentation. Okay, pretty much anybody except for strict aerobes are not going to do anaerobic respiration. So here's a key point. What's the difference now between anaerobic respiration and fermentation? It really depends on the fate of pyruvate. Okay, what happens to pyruvate? That's like a huge branch point. Or pyruvic acid, same thing. If there is, I'll go ahead to back to this overview. If there's an electron acceptor available, then those pyruvates will go into the Krebs cycle, eventually into electron transport chain. If there is no suitable electron acceptor, then pyruvates have no choice but to enter into some fermentative pathway. Okay, so I wrote down sort of the dictionary definition of fermentation here. With a fermentative pathway, important point here, the final electron acceptor is going to be some organic molecule that's already present in the cell. Okay, so most often that final electron acceptor is going to be pyruvic acid. Okay. And here's the other really important thing. What's happening here, the NADH is that come off of glycolysis, they are cycling back. They're dropping off their electron, usually with pyruvic acid is accepting an electron, and the NADH is oxidized back to NAD. And that's really important because if those NADHs coming off of glycolysis have nowhere to dump off their electrons, then eventually glycolysis will stop. Because if there's not that reduction from NAD to NADH, then that oxidation in the pathway can't happen. Okay, remember with every reduction, there has to be an oxidation. So if you don't have anywhere to dump off those electrons, even glycolysis will stop. Okay, so as we'll see, usually with fermentative pathways, we'll say, okay, let me write the NAD. So NADH oxidized back to NAD in a fermentative pathway. And the other hallmark of fermentation, usually there's going to be no additional ATP made. There's some exceptions to that. But basically, again, an organism is going through some additional fermentative pathway after glycolysis just to keep glycolysis going, just so it can get those 4 ATP by substrate level phosphorylation during glycolysis. Okay, so, one more thing. I'll write it here because there's room. From a fermentative pathway, bacteria are usually going to have acidic end products. Okay, so that will be obvious here as we look at some sample fermentative pathways. There's a lot of different fermentative pathways. This figure shows 2 possible fermentations, probably the 2 most famous. Okay, so here's the pyruvic acid. We get 2 of them when glucose is used in glycolysis. And this is a little bit confusing. An organism will either take one fermentative pathway or another. So this makes it look like because there's two pathways, there's some choice. No, it's got enzymes to do one thing or the enzymes to do another. Okay, so here's the simplest pathway, is lactic acid fermentation. Okay, with that kind of fermentation, the NADH, those electrons from NADH are dropped off at pyruvic acid. And the difference between pyruvic acid here and lactic acid is right here. Okay, there's 2 electrons and a proton there that are dropped off. And that's the only difference between pyruvic acid and lactic acid. Okay, so that's the simplest fermentation pathway. Again, you've got an acidic end product. The cell just can't get any more energy out of that molecule without a different electron acceptor. Question? So there are many other fermentative pathways. Here's 2 of the most common ones. I'm going to show you some other ones in a minute. Yeah. So it just depends on what enzymes the cell can make. This other pathway over here, where ethanol is an end product, it's an alcoholic fermentation. Okay, that's yeast that are doing that one. Okay, we'll say over here, some bacteria. Okay, certain bacteria can do that. Most bacteria can't do alcoholic fermentations. Okay, that's mostly yeasts are doing that. You'll see there's a gas coming off here. It's oftentimes carbon dioxide during fermentations. That's very common to get some byproducts of some gas during fermentation, but not the lactic acid one. That's just, again, that's why I called it the simplest one. We actually can do some fermentation in our muscle cells. If you've ever run a 400-meter dash, around 300-meter mark, that lactic acid is building up in your muscles and you're finding it really hard to keep moving. Okay, so we can do that on a limited basis in our muscles, but can't do it for very long. Right? Let's see. Okay, so I guess the main thing here, again, that these electrons are accepted, and then the NAD will go back, to glycolysis and keep glycolysis going. With either of these pathways, do you see any ATP produced? Nope, cell's just getting what it can get from glycolysis and not anything additional. I wanted to show you this. Actually, let me come back to this. Let me show you this picture. A week from, no. It's Wednesday or Thursday of this week. If you're in lab, you'll be inoculating these tubes that we call fermentation tubes. Sometimes they're called purple broths because they're purple when we inoculate them. But they're sets of 4 tubes. Each one of them has a different sugar in it, like glucose or lactose or mannitol. And there's a pH indicator in there. So we're trying to understand if the organisms have the enzyme in order to use that sugar. Okay, some organisms can use certain sugars, some organisms can use other sugars. It depends what enzymes that they have. And so we're testing if an organism can ferment a sugar, then you always get acidic end products, and there's a pH indicator in there that will change the color. All right, so for these tubes, the yellow tubes, yes, the organism could metabolize those because the pH came way down. The purple tube there, pH didn't change, so that organism couldn't use that sugar. So that's coming in lab. And with these tubes, we're not really doing anything to keep oxygen out of those tubes. Reality is that these organisms, if they're facultative, they're probably doing some aerobic respiration at the same time they're fermenting the sugar. Okay. I didn't post this because I didn't want anybody to worry that they had to learn all of it. But the point that I'm making here is that there are many different fermentation pathways, and it depends on the organism. It depends what enzyme they have. one of my favorite fermentation pathways is carried out by these Propriani bacteria. The end product that they end up with, makes sense, is propionate, and carbon dioxide is given off. Propionate is the molecule that gives Swiss cheese its really distinctive flavor. And those holes in Swiss cheese are from the carbon dioxide that's released during that fermentation process. So that's always been one of my favorite. Sometimes we can test to see which pathway a bacteria is using, and that helps us to identify the organism. So later on in the semester in lab, you're going to test to see if an organism is doing what we call a mixed acid fermentation, where acetic acid is one of the end products, or it's doing this 2,3-butane-diol fermentation. Again, that helps us tell certain organisms apart from other ones. Here's another cool one, the Clostridia bacteria, Clostridium. Hey, I know we talked about them in here as being organisms that can sporulate, that can produce endospores. Some of you have worked with patients maybe, or maybe you've had C. diff yourself. I've talked to college students that have had Clostridium difficile. That organism, when it ferments, it makes this intermediate butyrate. That is that's what's responsible for the whiff of the diff. That is extremely smelly intermediate that some people say they can identify, you know, diarrhea, a patient that has C. diff by the smell of their diarrhea. So some really smelly things associated with bacterial fermentation. I've read that the whole deodorant industry is based on bacterial fermentation. Bacteria like warm, moist places, like armpits, like feet, and they produce these, you know, really smelly end products when they ferment. So fermentation, it's a cool subject to think about. Okay, but recognize that because bacteria can ferment different products, it's been enormously helpful to us as humans. We get things that we eat, like Swiss cheese, things like yogurt, soy sauce, even as a result of bacterial fermentation. Of course, alcoholic beverages, that's the yeast, the Saccharomyces cerevisiae, things like that you don't even think about being, you know, products of anything organic, acetone, isopropanol, acetic acid, that's vinegar. So again, thanks to bacteria, not a list to memorize, just that, you know, recognize the help that they give to us. I want to end, so this is just one of those tables that can be helpful to kind of cover up and quiz yourself. Okay, are you getting the big picture here? And again, I'm not too worried about counting numbers of ATP or this or that, but that could be helpful. I want to finish here with talking about, oh no, we have a little bit more time. We're not quite done yet. So I want to finish metabolism with talking about and recognizing that most bacteria can use other things besides sugars for metabolism. Okay, they often can use proteins. They can also often use lipids. Okay, so just kind of thinking about differences when a protein is used to catabolize instead of a sugar. Remember, proteins are big molecules. They're polypeptides. They're made up of many different amino acids. And that's going to be a big molecule. Remember, bacteria can't phagocytize a big molecule to use it for nutrients because they have that stiff cell wall outside of their cell membrane. Okay, so they can't do endocytosis, but what they can do is secrete enzymes to break larger molecules down into smaller ones. Okay, and you're going to do a lab testing for these, what we call them exoenzymes, enzymes that are secreted outside of the cell, in a couple of weeks. And one of the kinds of enzymes that you're going to test for are proteases. So protease, that's just a general term for an enzyme that will break down a protein. All right, so it will break these peptide bonds between the amino acids. Okay, and now those amino acids are small enough that they can be easily transported into the cell. How are they broken down once they're inside the bacterial cell? There's some differences, but most often the organism will produce a deaminase enzyme, which does what it sounds like. It's going to cleave off the amino group of those amino acids. So what you're going to be left with, and remember R, that's just the variable group, whatever difference depending on what amino acid. But what you're left with is this some 2-carbon compound. And remember, where 2 carbon compounds are accepted, they can go right into the Krebs cycle. Okay, they'll combine with coenzyme A and enter right into the Krebs cycle. So you don't get, you know, the substrate phosphorylation or the electrons coming off of glycolysis. They enter right into the Krebs cycle. And this amino group, what happens to it? It will usually become ammonia in the cell. And what do you know about the pH of ammonia? Is it acidic or alkaline? It's very alkaline, yes. So if an organism is metabolizing proteins, you usually get alkaline end products. So it's metabolizing sugars, acidic end products, metabolizing protein, alkaline. What about lipids? Okay, lots of energy potentially for bacteria for metabolizing lipids. Lipids are just composed of 3 fatty acids and this 3-carbon glycerol. Okay, so once again, the cell will secrete some enzyme extracellularly, like lipase. So an enzymes they're going to test for in lab in a week or two. And that will split the lipid into fatty acid chains and glycerol. So here we see that here. Here's the 3-carbon glycerol. Okay, the lipase is going to cleave the molecule right there, and you end up with glycerol and 3 fatty acids. So first of all, what happens with that glycerol? A little ATP has to be added in, but then we get this dihydroxyacetone phosphate. Okay, and if you remember, okay, this was the slide that we used before. We were talking about glycolysis. That's one of the molecules that results when you split the fructose 1,6 biphosphate. Okay, and I talked about how that's not a symmetric molecule, that usually that dihydroxyacetone phosphate gets converted into glyceraldehyde 3-phosphate. Okay, so that's where that enters into glycolysis. So we get quite a bit of ATP, quite a bit of electrons just from that 3-carbon glycerol. What about the fatty acids? Okay, that's shown over here by a process called beta oxidation. Okay, then there's some coenzyme A's that are attached, and just by that process, like that transition step from pyruvic acid to acetyl-CoA, we get some reducing power out of that. And then this beta oxidation, this 2nd carbon from the end, that's the beta carbon. Okay, so basically we get cuts made every 2 carbons here like this, and each one of those 2 carbons becomes an acetyl-CoA. So all of those are entering in to the Krebs cycle. Remember, like right here. Okay, so we're getting lots and lots of electrons coming off each turn of that Krebs cycle. So those fatty acids be converted into a huge amount of energy. Again, that's the process of beta oxidation, kind of in a nutshell. Okay. All right, questions? Yeah. So can you make acetyl-CoA without the oxidation of fatty acids? Yes. So when, remember there was a transition step with pyruvic acid where it loses a carbon to carbon dioxide and the acetyl-CoA. Apparently acetyl-CoA or coenzyme A is necessary to kind of enter the acetate into the Krebs cycle. But yeah, it can start out as a sugar, as glucose, or it can start out as a fat. I think I have one more question here before we move on. Okay, one more review question from today, and then we'll start our genetics unit here. What do you think? Oh, I'm seeing a hand signal for B. Yes, okay, so B's correct answer. The other three answers, those all describe fermentation. So a lot of times, easy test questions for me to make are, in what pathway does this happen? In what pathway does this happen? Okay, so kind of know the difference between fermentation and anaerobic respiration. What happens, you know, in the Krebs cycle, what happens, electron transport chain, things like that. Okay. Okay, deep breath. Moving on. Okay, another big unit here that we're going to spend some time on our genetics unit. So OpenStax kind of splits this up into two chapters. Chapter 10 is biochemistry of the genome. So your book spends a lot of time going through some history. We're going to pick out a few things there to talk about, and then thinking about the structure of DNA and RNA, and hopefully that will look pretty familiar to you, as well as kind of the differences between a prokaryotic genome and a eukaryotic genome. Again, that we've talked about a little bit already, so that should be a little review as well. All right, so the first thing that your book talks about is, it seems like at first kind of a weird place to start, but in terms of our understanding of what genetic material is, this was kind of a big step, an experiment that now we understand was really important. And this was an experiment by Frederick Griffith. And he published this experiment in 1928. So if you think about, at that time, we kind of knew that there was something that functioned as a hereditary molecule. We didn't really know what that molecule was. We certainly didn't know the structure of it. Watson and Crick's paper wasn't until 1953. So again, we knew that cells, you know, had something going on. And studying the process in bacteria made sense because bacteria are simpler. Jacques Monod is quoted as saying, "What's true for the elephant is true for E. coli." And there's a lot of truth to that. Again, we're going to study bacteria because things are a little simpler. We will be pointing out differences between a lot of processes in eukaryotes and prokaryotes, but there's a lot of truth to that as well. Okay, so what he did in this experiment, he took, Frederick Griffith took 2 different strains of Streptococcus pneumonia. That's an organism, we'll talk more about Streptococcus later on in the class, very famously causes a really severe pneumonia. And he took 2 different strains, the S strain, and a strain of a bacterial species is just kind of a little variation of a species. So the S strain, that was what we call the wild type. Okay, it made a nice capsule. Okay, so remember one of the functions of bacterial capsule helps a bacteria kind of establish itself as an infection in an organism because it kind of keeps our innate immune cells from getting after it right away. So again, it helps it keep it from being phagocytized. So that S strain made a really nice capsule. And then the R, or sometimes we call the S strain the smooth strain. When you see the colonies of a capsule producing organism, they look kind of slimy and smooth. And then the R strain, or the rough strain, this was the mutant. And this was the strain that, sorry, I didn't leave myself much room here, could not make the capsule. So when he injected that rough strain into the mice, nothing happened. I don't even know if the mouse got sick. That strep pneumonia couldn't establish itself in the lungs of that organism. And maybe what will help you understand here, when he injected that S strain into mice, the mice would die. Okay, so the wild type strep pneumonia, you give those mice a good enough dose of that and they'll die from the infection. So then he did kind of a strange thing, and I don't know what made him think of doing this, but he heat killed that smooth strain. So he took those virulent, those disease-causing smooth strain, killed them with heat, and then injected those dead cells into the mice. And the mouse lived like you expect it to, because the bacteria were dead. But then he took some rough strain mice and the dead heat-killed smooth strain. And here is the really interesting thing, the mice died. So separately, the killed smooth strain and the rough strain separately, neither one of them harmed the cell, but when you mix those together, then that causes the mouse to die. So what was going on here? Basically, some of the DNA from that smooth strain that was dead was able to leave that cell because the cell walls were breaking down and enter in the live rough strain and change that rough strain into now being able to make capsules. Okay, so this was evidence for something that he termed transformation. Sorry, ran out of room there. Okay. And they talk about that a little bit in the caption there as well. Okay, so this was a kind of what we call now horizontal gene transfer, and we'll talk more about this later. But this is something that bacteria can do. They can transfer some of their DNA from one cell to another in the same generation, not just vertically, like parent cell to mother cell. Okay, so there was some, hereditary principle, again, that was leaving one cell and entering another. There's evidence for that. But he didn't know like what exactly it was that was being transferred. So about 20 years later, there was another experiment kind of piggybacked on top of Griffith's experiment that helped us understand what exactly that hereditary molecule was. And this was an experiment done by Avery, McLeod, and McCarty. So got the names over there. And again, this was in 1944. And apparently we knew of the molecule of DNA, we just didn't understand the importance of it. So what they did, again, they took the killed S strain from strep pneumonia. They somehow removed those, the capsule from that, because they knew that that didn't play a role in the actual hereditary principle. And they didn't really understand if it was a protein, if it was DNA, or if it was RNA that was the main hereditary principle. They talk about this in your book, that they really thought that it was protein was the most likely candidate, because proteins are made up of 20 different amino acids. There's a lot of, kind of variability there. So they thought that that was probably a protein, but they didn't know for sure. So with this experiment, they took those components, and when they didn't do anything to it, and they added the rough cells changed into the smooth cells that could make a capsule. Okay, so they understood that. And then to try to figure out if it was protein, RNA, or DNA, they added protease to one tube. So degraded the proteins, okay? But still, after they added the R cells, they transformed into smooth cells. Okay, so it wasn't protein because they got rid of all the proteins. With another tube, they added ribonuclease to degrade RNA. So there was still protein and DNA in there. And when they added the R cells, that transformed as well. So they knew it wasn't the RNA. And then when they added enzymes to degrade DNA, now there was no transformation. So they concluded, oh, this unit of heredity must be DNA. And then it was only a few years later that we elucidated the structure of it. That was Watson and Crick's paper. Okay, so structure of DNA. Hopefully this looks somewhat familiar. There's going to be a sugar. Okay, and this is a 5-carbon, we call that a pentose sugar. So deoxyribose, that's a sugar. It's going to be bonded to some base. We'll look at the different bases in a minute, nitrogenous rich bases, and a phosphate. Okay, so the three of those things together make up, we call that a nucleotide. Okay, the sugar, the base, and the phosphate, that's a nucleotide. And if you look a little bit closer at this deoxyribose sugar, when you start at the oxygen there and you go clockwise, you call this the 1' carbon, the 2' carbon, the 3' carbon, 4', and then the 5' carbon is not part of that ring structure. It's sticking out from it. Okay, so this is how we get the directionality of DNA. This is the 5' kind of side of it. And over here, this side would be the 3'. And that makes more sense when you have many in a row. Okay. So here's the bases. Again, we're not need to memorize structures or anything like that. The cytosine and thymine are considered pyrimidine bases, and the adenine and guanine are the purines. And they also have 2 rings. I had a little mnemonic that helped me remember that when I was in college. My major was part of the ag school. And so I always said Aggies, so our adenine and guanine are 2 pure. Okay, two rings are the purines. So I don't know, that's how I remember that. Of course, if, well, we'll move on to the next slide here. Okay, so here, when you have 3 nucleotides bonded to each other, now you get a sense of the directionality here a little bit. This 3' hydroxyl group at one end, and here's the 5' carbon at the other end. Okay, there is a kind of a directionality to this, as we'll see. Each nucleotide is bonded to the next nucleotide with a a phosphodiester bond. Okay, this is a kind of covalent bond. And these are the strongest bonds in nature because the electrons are shared. One source said there is no conflict to weaken the arrangements. Okay, electrons equally shared, that makes for a very strong bond. Now there are other bonds in the DNA molecule. There are hydrogen bonds. So here DNA is double-stranded, right? And we have here there's 2 hydrogen bonds that will link one strand with its complementary strand between the adenines and the thymines. And then here between the cytosines and the guanines, there are 3 hydrogen bonds. And that becomes important in areas where the DNA is going to unzip. We usually see a stretch of A's and T's, okay? Those hydrogen bonds are gonna be, there's only 2 of them, those are broken a little bit easier than if there are 3. And I should point out, if we know, there's a homework question about this. So there's something called Shagriff's rules of pairing, right? The adenines are always paired with the thymines and the guanines always paired with the cytosines. So if we know, say 20% of the bases in the cell are adenines, and we can tell how much percentage of all the other bases there are, because there's always going to be the same number of thymines as adenines. So 20% would be thymines, and then that makes 40%. That means the guanines and the cytosines are going to add up to 60%. So there's going to be 30% C's and 30% G's. Whoops. Yeah. So I mentioned that DNA is double-stranded. What is the exception to that? Where in nature do we find single-stranded DNA? Anybody have any thoughts on that? We haven't gotten into that. We haven't talked about that yet. What do you think? What's that? Even in gametes, it's double-stranded. What do you think? Well, so DNA isn't, it's in the nucleoid region of a bacterial cell. Not really, I suppose maybe there's a possibility that it could be damaged. What are you thinking? Oh, so ribosomal RNA, yes, but that's RNA. Oh, viruses. Yes, yes. So there are single-stranded DNA viruses. Okay, so that's good. We haven't talked about that yet. There are also double-stranded RNA viruses, which we usually don't see RNA double-stranded. So that's the difference. So, okay, so double-stranded, double helix, that was elucidated by Watson and Crick, not because they did a bunch of experiments to figure out the structure there, but they just looked at everybody else's experiments and kind of figured it out from everybody else's. The one other thing to mention here, that those 2 strands of DNA are anti-parallel. Okay, so here we've got the 5' end of this strand and the 3' end. The other strand you'll see is oriented the other way. There's the 3' hydroxyl group, and here's that 5' carbon. So they are always oriented in the opposite direction. Okay. Good. So differences between RNA, there's really not a lot of differences. Of course, there's going to be ribose sugar instead of deoxyribose. Look at the structure there. They are so similar. The only difference here is ribose has a hydroxyl group there instead of just a hydrogen. And incredibly enough, that weakens the structure of ribose just a little bit. So when we find RNA in the cell, it's usually fairly temporary, with the exception of ribosomal RNA, messenger RNA or tRNA. Those are all just kind of temporary structures. And of course, we have the base uracil instead of thymine as part of RNA. Okay, that seems like a good place to stop for today. So we'll carry on with that. We'll probably get started with replication on Wednesday.